Okay, so welcome and thank you for those who have joined us in person and those who are joining online via Zoom. I am happy to do the December Biotech Tech Talk. The title of my talk is Improving Rigor and Reproducibility by Preserving Valuable Rodent Models. I want to announce that in January, 10x Genomics will be hosting the January Tech Talk, and that is going to be facilitated by Sandra Splinter-Bondurant. And their title is Biology of True Resolution, a live demo of 10x genomic data software. And with that, I think I'll just start. So for those of you that are online, feel free to add questions to the chat, and then I'll answer questions all at the end, and Liz will help moderate those questions at the end. Okay, for those of you that don't know me, I'm Kathy Krentz. I run the Animal Models Core. I've been here in the Biotech Center for quite a few years now, and I'm hoping you'll learn a few things today, as well as the big announcement, the big reveal of our sharing program that we have now up and running to a small extent. And then hopefully in the new year, we'll really expand it and get it going. Okay, so the plan for my talk today. I'm going to give you a little history about the cryopreservation program that I oversee, how we do it, what we do, kind of what we did over the course of this year, some fun things that we were able to accomplish. I'm going to do a tutorial on our software, our inventory system, which is an online system in FreezerWorks, and I'll talk extensively about the FreezerWorks program that we use. And then, as I alluded to, we are going to introduce the Wisconsin Rodent Sharing Program. And I'm going to talk all about that in that website that we have beta testing at this time. And then we're going to end with a short webinar from Jackson Labs. So to just overview how we do some of our cryopreservation work that we do for sperm cryo, it's important for us to know, pay attention to the morphology of the sperm. So the rat sperm that we work with periodically, the head of the sperm is a very sharp hook. And that head of the sperm contains all of the nucleus, which has all of the genetic material. And so it's important during the freezing and thawing process that we keep that head intact so we don't damage the genetic material. There's a mid piece that connects the head to the tail, and that mid piece just serves the purpose of holding things in place, as well as it's important during motility. And then there's the tail. The tail can vary in length, different lengths from mice to rat even. But the tail is, of course, the main reason or the main mechanism that the sperm gets to an egg. But sometimes we can even have different procedures if the tail is damaged during the freezing and thawing process. We use a universal freezing method that was published a few years ago. And so the universal protocol that we use is used internationally. And so that's kind of important when we're shipping things all over the place, that everybody at least is aware of the different protocols in place, so you know how to thaw the sperm, because that's very critical when you're doing an IVF experiment to recover live offspring. I think it's important for everybody to realize in the methods we use to freeze down the sperm, we use these plastic straws. So these are roughly 5 inches in length. There's a cotton plug that in the center of that is a powder, a PVA powder. And then what happens is we can move that up and down the plastic straw, but hook this straw to a syringe, and then we load the liquid sperm up into the opposite end of the straw. We load some media that hits the cotton plug and actually forms a gel, and that will prevent any movement within the straw. And then these two small pieces of liquid down here are the actual samples of sperm. So we load 10 microliters of sperm per straw. And so we can get roughly 16 samples or aliquots per line. And therefore, we have a whole bunch of aliquots that we can then store in-house, or we can ship them off to a collaborator. Every time we do a sperm cryo, we do an analysis on our computer system. So this microscope, we load a small sample. It will color code the sperm, either green or blue or red, depending upon if it's moving in a straightforward fashion, whether it's spinning in circles, or whether it's dead. And so then the computer captures all of these snapshots across the microscope slide, and then it gives us a readout as to how many are modal and how many are moving perfectly in line, and then how many are actually dead. And so we use these percentages in this far right column as a guide to pre-freezing parameters so that we could tell a client whether their samples are good to go or not. Because sometimes we do see motility way down in the 20% or even 10%, and that's not good enough for us to guarantee that we can bring these back after thawing. So all of this data is stored on lab archives. We now started doing that during 2025. We share that program or that file with each client when we do that work. And every once in a while, we do a random control thaw. And so we take an aliquot of sperm with the poorest motility, and then we do a IVF with that. And we figured if we can bring back a line that has pretty poor motility, all of the other stuff we should be able to easily bring back. So that's how I run my quality control within a cryopreservation a day or experiment. Okay. For embryo cryo, it's a very different procedure. So for mice, we have to do IVF. So in vitro fertilization, we take oocytes from a bunch of young females. We can collect sperm from one male, or we can thaw a frozen aliquot of sperm. We do IVF, in which we culture the sperm and the eggs together. We wash off the excess sperm, do overnight culture. And then in the morning, we have 2-cell embryos. These 2-cell embryos, we then put into a programmable freezer, which will slowly freeze the embryos down to -35 degrees C. And from there, we can put them into one of our liquid nitrogen tanks. So all of our samples are divided into 2 different tanks. And we do that as, again, a quality control, so that if something happens to one of the tanks, the other tank should be fine and your samples should still be preserved. This is an inside picture of a liquid nitrogen tank. The tanks have these canisters, if you can kind of envision this. There's 6 different canisters, and we can pull one of these up, and then down inside of the vapor phase are all of these canes that hold the straws. And this is the way we can maximize our space. Instead of freezing in vials and using boxes, this allows us to freeze thousands of more samples within one given tank. We also color code the tops, and that we record, again, in lab archives and in freezer work. So we can easily identify certain lines by the color that the top of the tab is. Okay. So all of our tanks that we have, I will admit they're all stored within this building. They are in one of 3 locations, and those tanks are all locked. We have keys that we keep in a separate drawer. They are all monitored by Sonic U, and that's the alarm system. Oh, I got a misspelled word there. Sonic U, which is an alarm system that the UW or SMPH has now implemented and helps support. All of these are inventoried in our freezer program called Freezer Works. I'll talk a little bit more about that in a little bit. We currently have 23,339 samples in Freezer Works. All of these samples, I can tell you exactly which tank they're in and in which position they are in within a tank. We currently have 18 liquid nitrogen tanks. And again, all of those are secured within the Biotechnology Center. So to bring back, to recover live animals for mice, we do IVF, in which we put the sperm and the eggs together in vitro, and then we can transfer those embryos into a recipient mom, and she can give birth to the pups. For rats, because of the length of the tail, rats we can't do IVF with, or not to the point where I'm happy with the results. So instead, we do ICSI, intracytoplasmic sperm injection. And Felipe, who is actually here in the audience, he is very good at this. You could take a rat oocyte, you remove the tail from the sperm, and you go in with this very fine needle, and you pick up just the head of the sperm, and then you physically go in and deposit the head into the cytoplasm of the egg. And then these divide, and again, the same process is the same, where we put these rat embryos into a recipient mom and we get pups born. So we are able to do ICSI and IVF within the Biotech Center. And then lastly, anything that's stored in our liquid nitrogen tanks, we can export to collaborators. And this is one of the big things I want to encourage people to do. And I'm going to tell you about our sharing program and how I hope to expedite or help expedite this process. Okay, so a little history about what we've been doing and how long we've been doing it. So I joined the lab, which used to be called the Transgenic Animal Facility, back in 1991. So over 30 years ago. We froze the first set of embryos in 91. We still have some of those embryos still in our tanks today. In 2006, we started freezing sperm, but we froze those in vials because at the time that was the best method to use. It worked, meaning you could get the mice back. It didn't work great. In 2010, Jackson Lab, as well as others, came out with some new sperm cryo protocols. And that is the basis of what we use today. It's been tweaked a little bit, but then we started freezing everything in straws. So these vials are in those big cryo tanks that hold boxes. The straws are all in smaller tanks. And so now that's all extensively what we use is plastic straws. We implemented a new cryo IVF recovery protocol in 2015. During COVID, in 2022, we merged all of our data into FreezerWorks. All of our data used to be in Excel, and that's just not a good way to have data. And so we merged everything into FreezerWorks, and labs are able to log into FreezerWorks and see what samples they have. And it actually, I think it's a great system to use these days. 2024, I said we brought back our first line via ICSI, and then in 2026, I'm hoping to implement this sharing program. Okay, so how old can we go? Okay, so 2 things we did this year. Paul Lambert's group out at McCardle or Oncology, he asked us to thaw a batch of embryos that were frozen in 2015. So that's 10 years that they've been hanging out in a liquid nitrogen tank. Those embryos we thawed, we did the embryo transfers. We have mice downstairs right now that are from that batch of embryos. An even better story, Chris Bradfield, again in McCardle, he had us thaw something this year. These were frozen embryos that were frozen in 2003. Frozen for 22 years. So number 1, we were able to find which tank they were in, and we were able to find them. Number 2, we got live offspring. Number 3, they genotyped correctly. So to me, that's a test to how good of a system we have in place. And how the methods that we used back then, we're able to figure out what we use, because that's also important, just to, as you change protocols, that you have to monitor what you did and at least make records as to how you did things so that you can reverse order that when you do the thawing and you know what's in these straws. Our longest record, the stuff we did for Michael Gould, Michael Gould stuff that we thawed back out in 2022. his samples were frozen for 25 years. So that's the longest we've done. Jackson Labs probably now has stuff that they've frozen for close to 40 years and that they have brought up. So again, I run a cryopreservation program that is, I think it's a really good program. We have a long history of success. And I don't think there's a reason why anyone needs to go off campus for these services. I know there are companies out there that you get emails from that say, send us your mice, we'll freeze down your animals for you. I don't believe you need to go off campus for those cryopreservation services. And we'd be happy to talk more if anybody has questions about whether a company can do things better than I can, or my staff can do them. Okay, so an off-campus client, we do some work for off-campus clients. We've had other institutions ship us males and we freeze them down here. We did do some rat sperm that I alluded to. This was the ICSI project, and then we thawed those back last year. So in order to enhance your research, improve rigor and reproducibility, I think there are times when you need to revert back to these frozen stocks. If you have genetic drift happening, if you've lost a phenotype, there again are reasons why we can go back to the frozen stocks and recover them, and then potentially get back a phenotype if you did happen to lose one. Okay, so now let's go on to see what else have we done during the course of this year. So this is importing. So this is stuff that came from off campus. So we got sperm from Germany, we got sperm from Canada, we got sperm from Sweden, and we got embryos from Japan. All of these, we were able to recover live offspring here at the UW without too much problem at all. And again, universal protocols that everybody shares within our technology, or the transgenic technology. And that makes things so much easier when facilities like mine share their protocols and have things in place for importing and exporting. Within the United States, we got stuff both from North Carolina and California. For those of you that aren't aware, MMRRC stands for the Mouse Mutant Resource and Research Center. This is an NIH-supported repository. There's 4 of them. Jackson Labs is one. There's North Carolina, Missouri, and then UC Davis. And that, again, is a repository similar to what we have here, but at a much higher scale. And you are able to donate your mice to these repositories. I don't know that they take everything, but again, it just depends on what the main purpose of sharing. But these repositories do exist within 3 separate locations. And so we do IVF, again, we do embryo transfers, and we can have the correct mice born. So these are just some examples of importation that happened over the course of the last year. Now in terms of exporting, so what have we shipped out of BICOR and where have we shipped it to? So this is for the last about a year and a half. So we sent sperm to Sweden. And this was done earlier this year. I won't say it was challenging, I won't lie, it was challenging due to quite a few different factors. So anything shipping out of the country has to have a USDA health certificate. That health certificate is only good for 10 days once it is sealed at the USDA office. So and then once you ship things, the dry shippers are only good for about 10 days. So you kind of have to have everything aligned, and you have to have a good shipping company that's going to make sure all of your documents are in place and that everything is properly filled out so that it can get through customs faster. So we were able to get this sperm to Sweden. They recovered live offspring, and they have mice there now using sperm that we froze here. The other hiccup became when my dry shipper was coming back It got held up in customs because it is made of metal and they wanted to charge me a tariff. So I had to pay the tariff before I could get my liquid nitrogen tank back, which is, again, you have to, everything changes within a given year depending upon certain conditions. So we now know all of these details, and we could easily send more samples internationally now. So within the United States, it's easy, much, much easier. So we have shipped sperm and embryos to Yale. We shipped live mice to Virginia Tech, and those were recovered from frozen stocks. We shipped sperm to Michigan. We shipped frozen embryos to UAB. We shipped live mice down to MD Anderson. And again, those were recovered from frozen stocks. So this sharing program we have been working on or doing, we just haven't broadcasted it as much as we're going to start doing in the next year. Other ways that MyCore can help with assisted reproduction and IVF or frozen stocks. So let's say your goal was to have 50 age-matched males and females. So let's hypothetically say you need 100 mice. You need 50 that are males, 50 that are females. You could set up all of those breeding pairs, and you could maybe get pregnancies, and you may have litters all born within a given time frame. I propose that we do IVF instead. If you can do IVF with either frozen or fresh sperm, we would need young females to serve as egg donors. We can do mass production through IVF, do all the embryo transfers within a day or two. You can get a large amount of mice born within a 1 or 2 day period. And this is something, again, that I would like to promote and encourage people to do. If you are looking for stuff that has been frozen down in particular, we can do create a cohort of animals within a small age range if needed. Other things that we have helped with, so fertility of mutant males. So some people have come to us and asked us to help figure out why their mutants aren't breeding, and if there's a phenotype within the sperm, and our computer analyzer will analyze different morphology structures. And so we're happy to talk about those types of experiments if you are interested. We've also analyzed some ovarian functions within some mutant females, if that is something that you're interested in as well. Okay, so now we're moving on to FreezerWorks. FreezerWorks is our online database. So this is a database that we share with the Reach Core. Reach used to be called Show. They're a repository for human samples. And so this is the website, it's within the School of Medicine server. And so because of that, you can only use this on UW campus, or you have to VPN in. But this is what the home page looks like. And within FreezerWorks, you can see that we have continually increased the number of samples we have frozen down. As of today, we're up almost 24,000 samples. That is inventoried within FreezerWorks. Okay, and so each year we charge a yearly storage fee, and that is based on the number of frozen samples that we have within FreezerWorks. So that's how we determine how many samples we have frozen. We figured out our yearly costs, and then we divide them per lab. Give me a second, sorry, I need to. Okay, sorry, I have to go to. Okay, so this is the website. And so I am going to log in. So I log in, and I have different credentials than other people do, but this is what you're going to see once you log in. And then all you do, we have set up each lab with their own account and their own password. And so everybody can log in under their own credentials. And then if you go to search and report and you run your saved search, I'm just going to pick one because I don't, I have access to everything. You're going to run your save search, and this has all been set up for you. And then down at the bottom, you just say execute search. And so this will show you all of your samples. So there are 3 tabs, one that says samples, one that says aliquots, and one that says transactions. So the samples, again, remember we share this with a core facility that stores human samples. So they have labeled this samples. I would like to call it line names, but I can't change that. So this sample tab will give you all of your line names, okay? And it will tell you how many aliquots you have frozen down. FreezerWorks gives them all a unique ID number. And a lot of this, so this creation date, this was when we merged things in. This is not the freeze date. This is a creation date. So this was all happening during COVID, and that's when we were merging data in. If I go to this aliquots tab, you will see every sample that you have. And so I picked Nader Shabani, and I hope he doesn't mind that I picked him. But his they go on and 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 on. And then you could go to the next page. So this is literally every sample that we have frozen down. And the unique thing about this field is it actually will tell you what liquid nitrogen tank it's in and what position. So if you remember that picture I have of the inside of a tank, there's 1 through 6. So tank 3, position 4 carries this particular line name. And so you can see tank 14, tank 16. You know, it tells us exactly where these samples are within our liquid nitrogen tanks. Again, it assigns a unique ID number, and that's per sample at this point. Okay. And then the last tab is called transactions. These are things that we have thawed. And you could see the line name, if we thawed it, what the date was that we thawed it, and then what did we use it for? You know, if we used it for an IVF or we thawed it or, you know, whatever, okay? So this is the data in FreezerWorks. This is what all labs have access to. Once a year when we send you that invoice for billing, that will include your login and your password, but we are happy to help you navigate this website. Because that's where you're going to see your data and all of the lines that you have frozen. Okay, now back to this. Okay, so onward. Now we're switching gears again. We're going to talk about what I have termed the Wisconsin Rodent Sharing Program. And I'm open to other ideas for what this can be termed or called. But just based on some other repository websites, this is what I've decided to call ours. So this is going to be an online listing of investigator strains that are stored within our repository that can be shared either within the UW or can be shared globally. PIs will have the option of only sharing within the UW, or they can share to the entire public. And the website will control what it lists. This is a completely voluntary program. I will be contacting each PI if they are willing to share their lines. If you haven't received an email yet, I did send out some emails already. You'll be receiving an email soon. And what we have done is we've created a Google Sheet of your lines that were listed in FreezerWorks. That Google Sheet will have all options for each line as to whether you want to share it. So it's on a case-by-case basis as to whether you want to share something or not. We have a built-in monitoring system within our spreadsheet, within our website, so that the total number of samples does not fall below a set point. We don't want to share all of your samples and then not have anything left. So we've built in some things so that we don't share everything. Okay, so this is what the website will be. You do not have access to it now. Right now, it's in beta testing. but it's called mouserepository.biotech.wis.edu. Now let's see. Okay. No, you can't see. Give me a second. Sorry. Okay, here we go. Okay, so here, this is what the website looks like now. Yeah, again, it's not live yet, but we are working on it. I have to thank the IT department within the Biotech Center because they have been instrumental in setting this up and are working with us to fully implement this. So the homepage will just have some introductory information. We're going to have both mice and rats within this repository. This is going to facilitate the compliance of the NIH sharing policy. Any funds that were any NIH funds that were used to create a mouse or a rat line, you're supposed to be able to share those, and this will help meet that requirement. And then at the bottom here, you see that there's a tab that says available mouse lines. So then if you click on that, you will see a list of lines that are currently shareable. So I appreciate Chris Bradfield, who went through his big long list, and he designated which ones he wanted to share. And this is the actual list of his lines that are currently in the database to share. A few things that I want to point out. The website will list whether we have frozen embryos or frozen sperm. There will be a publication link, if applicable, to any particular line. These middle columns, the total number of aliquots, the freeze date, and the freezer work ID, these three columns will be hidden on the website. That information is for my staff. You can see that this top one only has 2 frozen aliquots. Well, we need to know that. We need to know that if anybody requests this particular line, that instead of giving this away, we probably need to recover this line and freeze it back down again prior to distributing this line. But other samples, see how there's 24 samples here, there's 16 here. We have plenty of samples most times that we can share your samples without impacting the stock that we have in-house, okay? And so again, it goes on and on. And so we'll, this is what the website is going to look like. And so once I get more feedback from more labs, and we'll just start inputting it into the database, the website, and then that will be live at some point in time in the near future. So a few things that we have: a search button that if somebody's looking for a particular genetic strain or gene line name, they can put that in and it will search appropriately. We will also have a request this mouse button, and that will take them to a form that they fill in. And then that form will send an email to my team and to the donating investigator. So the donating investigator will know anytime their strains have been requested. This will be, I want to be open and honest about who we're giving stuff to. And that will hopefully, that's going to all be set up in the very near future. And then if you scroll down, the other thing I hope to do is I hope that within a strain that we can expand, like if they tap on this strain, this will open up and we'll have more information on this strain as to how it was made, whether it was made by targeted ES cells, whether it was made by CRISPR, what's the background strain? Was it, what was it originally created on? Is this now on a, what purebred background? So I'm hoping to add more data to this. Again, this is just an early phase of what we're going to be doing. But I'm really hoping that in the year 2026, we can expand upon this website and have all different lines from multiple investigators. Okay, so again, the website's built, it's not live yet, so don't go there, you won't get anywhere. But we'll let you know when it is ready to go live. So I just included this in case I had a major fail. Okay, so a few things about the website and our sharing program. Off-campus requests will require an MTA. I've talked to tech transfer people on campus. They have a simplified format, and that's all been established, and hoping that that can be expedited once we do get requests coming in, that MTAs can be completed fairly quickly. I've already told you about the request this mouse button that will send an email to my core as well as the donating PI. There will be a distribution fee with the option to either ship the frozen material to the requesting lab, or my core can recover that and ship live mice. So it just depends on what the requesting lab is looking for and what they have the capabilities of doing in their own laboratory or university. The distribution fee will be a fee that my core can recover some funds that we've been incurring to try to develop this website, to all of the time that is being spent sending emails and getting keeping the website up and running. It can also include a payment to the donating lab if the donating lab would require that. And so again, everything is going to be on a case-by-case basis as to how labs want to donate their particular lines and any regulations that they want to set up within that. Okay, so the last 5-10 minutes or so, I'd like to at least inform you about Jackson Labs website. Jackson Labs also does education, and aside from all of the mouse work and mouse development and purchasing and shipping of mice, they have a great education website. And I found this webinar, it was just put up in September 30th of this year, so it's only about 2 years old. And the title, Secure Your Genetically Engineered Mouse Models, Proven Strategies for Risk-Resistant, Resilient, Cost-Effective Colony Management. So this webinar is again available, but what I'm going to do is just play a small part of it. And it really follows what we've been talking about during this. So hopefully, I'm just going to play about 5-10 minutes of it. And so it's kind of in the middle of it, but it's talking about freezing down mouse lines. Why do you want to do this? When do you decide to freeze things down? And I thought it would be best that Jackson Labs can probably explain this a little bit better than I can.
Today, tomorrow, and years later through strain protective risk mitigation strategies, which will be the next big focus of today's presentation. One key part of that is managing your colony in a way that balances scientific needs with resource efficiency, which is something we'll explore next. Let's start with one of the most practical aspects of strain protection, cost management. Even with a well-managed colony, expenses can add up quickly. So here are some strategies to help you keep your colony efficient and sustainable. To keep your costs down, consider sizing your colony for what you have going on now, not what you think you might need 2 years from now. Colony sizes can always be increased or decreased with just a bit of planning if you decide your needs change in the future. Develop an accurate representation of your experimental needs. For instance, looking at similar published studies can give you a good idea of typical cohort sizes. Depending on your research field, you can also consider alternative ages or sexes. For example, depending on what you need, sometimes you may have performed only your experiments in males, but perhaps using males and females will yield equally important information and maximize the ability to use more mice from your colony for experiments. You can also consider, as I mentioned earlier, ordering in a cohort of mice for a specific experiment rather than committing the resources to build your own colony. And lastly, for strains that are in low use and that you don't think you are likely to use in the next 6 to 12 months or so, it might be worth considering cryopreserving your strain. And now this is something I'd like to spend a little bit more time on and explain why this is a really great strategy for saving your strain as well as resources. You may be wondering, what exactly is cryopreservation? Cryopreservation is the use of very low temperatures to preserve structurally intact living cells and tissues. In our case, we mean the preservation of mouse sperm and embryos. Now, effective cryopreservation isn't just about freezing cells, though. It's about freezing them in such a way which produces viable cells moving forward when thawed. Now, there are many reasons for why cryopreserving a strain is a great strategy to consider when a strain is low in usage. For example, if the strain is special but not actively being used, cryopreserving sperm or embryos helps prevent the loss of the strain. Additionally, the more lines are preserved as frozen stock, the less space they are taking in your colony room. To highlight this a bit more, let's go over this very quick example. In this left diagram, you see that there's a proportion of colonies, such as, let's say, this yellow dots that are inbred strains, that are perhaps less used as compared to the proportion of this hypothetical line, like this darker dot. However, when it comes to space that they may be taking up in the colony, the yellow colonies are taking up more space or caging than these other colony line. And let's say in this hypothetical colony, we even have extra legacy colonies, so-called. These so-called legacy or blue strains are from older studies that are merely being kept active. And while they're not being actively used, they are still taking up quite an amount of space. Over time, these strains can be more of a burden to maintain as more resources are being used to keep them live off the shelf, which can affect using funds for other purposes in your research. And one solution to this would be to cryopreserve those strains. Removing those lines, removing those blue lines, for example, could free up capacity, which could then support larger studies, free up labor, thus reducing costs. And you might be thinking, what exactly are those costs that you're referring to? Maintaining a line really doesn't cost that much, does it? In reality, there are a couple of costs with maintaining a live mouse colony that can be offset by freezing the strain. These could include space constraints, per diem charges, time, genotyping, and even labor hours. Besides, maintaining live mice keeps only a fraction of your strains available, but with cryopreservation, you can safeguard more strains without the ongoing cost of live maintenance. Even so, cryopreservation still remains underutilized. Now, most of the time when folks decide to cryopreserve a strain, it's usually at study completion or when the strain is no longer to be used. Cryopreservation isn't really thought about as a preventative measure for protecting strains like from breeding cessation or natural disasters. And so it's easy to fall into the thinking that this won't happen to me, or I have time, or I don't need to act now. But as this diagram illustrates, sudden situations can catch even the most prepared investigators off guard. Before 2020, few institutions prioritized cryopreservation because most had the resources to maintain live colonies. But when the pandemic hit, staffing became limited and maintaining live colonies became more difficult, which led to a surge in requests. Takeaway here is that while immediate threats often highlight the importance of protecting your research models, waiting for a crisis isn't a strategy. Proactive preservation ensures your research remains secure, regardless of when or if unexpected challenges arise. Again, risk assessment and management are critical. Now, in this slide, I want to show why being proactive about mitigating colony risk is so important by presenting some scenarios that vary in probability of occurring, as well as potential for impacting research. Here, the y-axis represents impact. And as we move up, the potential negative impact on research integrity increases. The x-axis represents probability. As we move right, the likelihood of a negative event occurring increases. The heat map colors help illustrate this as well. Cooler colors and greens indicate low impact, while warmer yellows, oranges, and reds indicate higher impact. For our first example, let's look at an event or a couple events that, although unlikely to happen often, have a severe impact when they do occur. For example, a natural disaster. Hurricanes, floods, fires, disease outbreaks may have, again, that low probability, but when they hit, the consequences they have can be devastating. For example, Tropical Storm Allison in 2001, Hurricane Katrina in 2005, and Hurricane Sandy in 2012 were detrimental to the research institutes in the affected areas. Millions of research dollars were lost, years of delay were caused due to project recovery, and worst of all, there was permanent loss of valuable mouse resources. On the other hand, there are everyday risks that occur more frequently. These include facility equipment failures, breeding errors or cessation, contamination, and genetic drift. When a colony is bred over many generations, more people tend to be involved in managing and caring for that strain. Without protective measures, even small mistakes like mislabeling cages or losing breeder records can accumulate. And each incident might have less impact than the natural disaster, but their higher probability means the overall risk to the colony can become significant over time. Now, for our next section, we are going to highlight how JAX can be your partner in protecting your strains. And for
Okay, that's where I'm going to stop the webinar. I just wanted to do that middle section because the last section is stuff that I've already told you about and how we cryopreserve sperm and embryos. And so that's already stuff that I have covered. And so, so with that, I want to thank my current staff. So this is a picture of my current staff, except for Kylie. Kylie was an undergrad student who worked in my lab and she graduated and we recently have a new undergrad. But Lucille, Felipe, and Jacob so this is Lucille, this is Felipe, and this is Jacob. They all do all of the cryopreservation that I've just talked about. Everything from the beginning where they collect the animals and freeze it down to recovery and shipping it off. I have to acknowledge my staff I had around COVID time. Hillary in particular was essential to taking the Excel data and merging it into the FreezerWorks program. And then my staff a while back, I say pre-COVID, that again did a lot of cryopreservation during that time. So I'm housed in the Biotechnology Center, which is under the Vice Chancellor's office. We are a shared resource within the Carbone Cancer Center, and we get some financial support, salary support from the Carbone Cancer Center. If you are a Cancer Center member, you receive a discount on labor. So make sure that you tell me that if you're going to have us do projects for you. And then we're also a core facility within the SFPH core directory. I listed my email address for myself and my lab, and then we have a website as well. So at this point, I'll take questions from either the audience or if there's any online, put them in the chat. Okay. Okay. Yes. Oh, yeah. Okay. So he asked basically, are all the samples from one strain in one liquid nitrogen tank? And no, we divide everything into two tanks. So if we freeze down a line of sperm right now, we get 16 aliquots. 8 would go in one tank, 8 would go in a different tank. One of those tanks is down the hallway from my lab. One tank stays in my lab, the other one goes down the hallway. So all samples, all lines are split between two tanks. So I think that answered his question. Uh, yes, yes, rabbit. Yeah, people ask me some weird things. Ground squirrels have come up too. But yes, rabbit is probably something, there was something I just read about rabbit. Okay. Yeah. Yeah. So we're going to have to work with tech transfer. And the MTAs, I'm not involved in them. That's going to happen behind the scenes between the donating investigator and the receiving institution or the receiving PI. So we will work out those details with MTAs. Okay. How about in the audience live? Anybody got any questions? Anything that, um, yeah, Jody. Yeah. You know, I have thought about that, and I thank you for raising that because I think that's a great thing that we could do, especially your core that has so many live mice in a breeding facility. Um, I'm starting off small because then you have to do the same thing. You'll have to get permission from all those different PIs. Can you share it? Who can you share it with? And yeah, and then you know, who pays for breeding up the colony and who pays? Yeah. But yes, I would love to have live animals be an option within the website. Okay. If nobody else has questions, thank you all for joining in person and online, and email me or let me know if you have any questions. Okay, thanks.